biological cell mass derived by abnormal and uncontrolled clonal expansion of single cell. So it is normal cell. There is transformation of this normal cell into malignant cell. And tumor immunology, why they found that patients with the immune suppressive drug or had primary or secondary immune deficiency more liable to development tumor. So there is a theory which is produced by Burnett and Thomas, which they demonstrate that there is a concept which is called immune surveillance. And this immune surveillance, that the immune cells, like macrophage, will continuously scan the blood and the body. When there is any abnormal cell presence, will be destroyed by these cells. And when there is defect in the immune system, so these abnormal cells will proliferate and develop tumor cells. And this concept will be approved by the patients with AIDS who had secondary immune deficiency develop Kaposi sarcoma of the skin, and patients with immune suppressive drug develop lymphoma. So we have two types of the antigen on any tumor cells, which is first tumor-specific transplantation antigen, which is mean that the antigen only presents on the cell wall of the tumor. And the second type of the antigen, which is a tumor-associated transplantation antigen, which is mean that these antigen expressed on the tumor cells are normal fetal cells, while in fetal, when this antigen presents during fetal life, while in the normal adult cells, they will disappear or present in low percent. And best example is alpha-fetoprotein, which is increased in hepatocellular carcinoma, and carcinoembryonic antigen, which is increased in colorectal carcinoma. And overexpressions of the these known as tumor markers. Tumor markers that they give a hint to the physician that there is a tumor when increase in its concentration in the body. And another type of TAT is overexpression of the protein or antigen on tumor cell, like epidermal growth factor, and they are normally expressed at low level on normal cells, while increased expression of these proteins, so this lead give a hint about the presence of the tumor. So tumor markers, either tumor antigens or tumor products, which is either enzymes or hormones, which is increase in its secretion in the serum. And these tumor markers used to diagnose the tumor or to follow up the response of the tumor to therapy. So immune response to tumor, non-specific immunity, which is the innate immunity, neutrophil, macrophage, NK cells. And if this fails, so there is a need to stimulate the specific immunity or adaptive immunity, which include the humoral immunity and the production of the antibodies by the B cell and complement activation, and also a cell-mediated immunity, which is a T cytotoxic cell and cytokine production like gamma interferon, interleukin 2, and macrophage activating factor MAC. The tumor also had a great mechanism to avoid the attack of the immune cells. And this include antigen modulation, antigen masking, sneaking through, tolerance, reduction of MSC class 1, lack of co-stimulatory signal, B7, CD28, and blocking antibodies. Stimulate the immune system to produce antibodies, which is blocking antibodies. That means without any benefit, block the immune response. Treatment of cancer or tumor: first, surgical resection of the mass. Second, send the patient to chemotherapy, radiotherapy, and immunotherapy used as adjuvant in the treatment of cancer to remove any tumor cells that evade the surgeon knife or chemotherapy or radiotherapy. This is achieved by vaccination. Vaccination is either killed tumor cells plus adjuvant injected to the patients, or used a purified tumor antigen as a vaccine. Third, using dendritic cells as a vaccine, and these dendritic cells pulsed with tumor antigen or transfected with plasmid expressed the tumor antigen. So this is the dendritic cells pulsed with tumor antigen. يعني نحقن بها الانتيجين, so it will express on the cell surface of this dendritic cells association with MSC. And this lead to stimulate the T cytotoxic cell, and this lead to activation of tumor specific T cells. يعني مو كل التي cell راح تصير بها proliferation, فقط اللي متخصصة للthis tumor antigen. And the second part is this dendritic cells transfected with a plasmid. This plasmid contains the antigen called the tumor antigen. This plasmid had genes. Sorry, this plasmid had gene encode the tumor antigen. So transfected, يعني يحقنوه transfected into the dendritic cells. So this lead to express the tumor antigen and this lead to stimulation of tumor specific T cells. Another type of treatment or vaccination using a biological response modifier like cytokines, tumor necrosis factor, interferon, BCG, dendritic cells, or T cytotoxic cells transfected with cytokine genes. يعني هناك قبل كانت transfected with genes encode a tumor antigen. هنا لا, transfected with genes encode cytokines. الماجيك بوليت, الطلقة السحرية, using monoclonal antibodies directed against tumor antigens coupled with diphtheria toxin or cytotoxic drug. 5. A cloned cytotoxic T cells specific for human tumor antigen. 6. Lymphocyte activating killer cells like tumor reactive cytotoxic T cell injected with interleukin 2, نسميها LAC cells. Lymphocyte activating killer cell. Another one, tumor infiltrated lymphocyte, يعني راح آخذ أني lymphocyte that infiltrate the tumor mass and re-injected with the interleukin 2 to the same patients, and it has a wide range of tumor activity and more toxic. So this is the tumor. This patient had a tumor, and this tumor infiltrated with lymphocyte, and I will isolate the lymphocyte from this tumor mass, and will proliferate in vitro and re-injected into the same patients with interleukin 2. They found that there is a tumor regression of the tumor regression in its mass and decrease in its size of the same patients. So this is called T cells, a tumor-infiltrated lymphocyte. While here in the lab, I isolate the lymphocyte from the blood and activate it in vitro and re-inject it to the same patients with interleukin 2. And this is another example, patients with CA lung, and I will took the lymphocyte which is present in this tumor mass, and then will proliferate them and then re-injected to the same patients with interleukin 2. And gene therapy nowadays use gene therapy like in COVID. Here use tumor cell or antigen presenting cell or tumor infiltrating lymphocyte and transfect it with B7 genes or genes encoding cytokines. And here we have the tumor cells transfected with genes using the plasmid. This plasmid had genes for interleukin 2 and B7, and this leads to continuous expression and stimulation to T lymphocyte. While here the tumor cell transfected with plasmid had genes for the granulocyte, monocyte, colony stimulating factor, and this leads to a stimulation of the dendritic cells. And by the way, these dendritic cells will lead to stimulation of the T lymphocyte. And thank you.